So I'm going to start with two numbers. Almost 40 million people are now living with HIV, and almost 30 million of them are on antiretroviral therapy. However, this antiretroviral therapy is able to reduce very nicely the replication in blood, but does not cure the infection. And also, the antiretroviral therapy does not fully restore the immune hyperactivation that people with HIV has, and also the immune exhaustion. As a consequence, HIV persists, and if they stop taking antiretroviral therapy, it's going to be a viral rebound. So people has to take antiretroviral therapy long life in order to control the viral replication. And this is because there is a latent HIV reservoir that is located in CD4 T cells that are the main targets for HIV. And those CD4 cells are distributed in different organs. As you can imagine, study those tissue reservoirs is very complicated because we don't have access to them. And this is why we started this project. This is a collaborative project that is funded by La Caixa. And here we wanted to study in the reservoirs that are in different tissues. And also for that, we established a collaboration with different departments in the hospital. This is a work that has been coordinated by Mary Cherjeneska. She is a PI in the Infectious Diseases lab too. And from our collaborators, we usually take some tissues, intestine, mainly colon, tonsils, and cervix. And what we do with that, this is the main objective of the project, is to develop tissue models of HIV persistence. So we want to develop those models in order to study first the different targets that we have in the different tissues, because we need to know them in order to design new strategies related to cure the infection. Also, we want to explore the innate immunity that are within tissues in order to develop those new strategies, immune-based strategies. So I'm going to focus on those two main objectives because we don't have time for more. So basically what we have is the tissues. We cut the tissues into very small pieces, and then we culture them into very small gelatin sponges. So the first thing that we did is to try to set up the model. So we characterize the CD4 cells, which are the main targets for the virus, that are in the different tissues. I'm going to show you the results for gut and for tonsils, because cervix, we are still analyzing the results. And basically, we did a flow cytometry panel, including different markers that allow us to characterize the CD4s in terms of their memory and differentiation, and also follicular helper CD4 cells. So this is the result that we have. This is a reduction of dimensionality analysis. And you can see here that the intestine and the tonsil, they have different population of CD4 cells. And those ones are the different clusters, meaning different agrupations of cells with similar markers. And if we check the differences, we can see that the gut, they have more clusters related with more differentiated CD4 T cells compared to tonsils that they have more naive CD4 cells and more central memory cells. So then we also infect those tissues in the lab. And this is how we monitor the infection. So we measure the viral antigen P24 within the CD4 cells. And this is the summary data for the different tissues, gut and tonsil. And we were wondering if the CD4 cells, they have different susceptibility to infection if they are coming from the different tissues. And the answer is yes. So the cells are differently susceptible to HIV infection. For example, in gut, we have more effector memory cells that they contribute more to the total infected cells compared to tonsils that they have more a central memory phenotype CD4 cells contributing more to all the infection. Also, in order to establish the latency model, we add antiretroviral therapy to our tissues. And we know the antiretroviral therapy induce latency. And this is what we observe. So basically, we reduce the levels of the P24 viral antigen. And also, in order to check latency, we reactivate those cells with PMA and Yanomycin, and we see a very nice reactivation, meaning that we have set up very nicely those models of HIV latency. So next, we want to assess the effects of latency reversal agents. Those latency reversal agents are different families of drugs that they are used for other applications. They are not specific for the virus, but the effect that they have is that they, for example, one of the families that has been tested in clinical trials are the HDAC inhibitors. Those drugs, they just open the chromatin, and then if HIV is inserted inside, it's going to be transcribed. So you are reactivating latency with those drugs. But as I said, they are not specific for HIV. So several clinical trials have shown that those allorays are able to reactivate HIV. However, they don't produce any reduction in the latent reservoir. And this is because two things. The immune system does not recognize and kill those viral reactivated cells, and also the allorays don't fully reactivate HIV in all CD4 cells. So we were wondering what's going on in the tissues. And for that, we add to our system model the allorays here. So we tested different families of allorays and also some combinations. And basically what we observe is that the two different tissues, they have different susceptibility to those allorays. For example, the SMAC mimetic AZD, it was much more effective at reactivating HIV in gut compared to tonsil. And on the other side, in general, also was able to reactivate most of the tonsil cells, but not the gut cells. So if we look at the individual clusters of cell population of CD4s, I'm not going to go into detail here, but just to show you that some drugs, they reactivate some of the clusters in one tissue, but not in the other tissue. So the summary, we have set up a tissue model of HIV persistent that recapitulates major attributes of viral infection and treatment. In both tonsillar and intestinal tissues, viral infection takes place in different populations of CD4 cells. However, how they contribute to the infection is different. Also, the action of different cure therapeutics on those CD4 cells and in the different tissues varies. So it means that the identification of those cell reservoirs in tissues is important in order to understand and to design those new drugs able or directed to try to cure the infection for HIV. So I'm going to finish with the acknowledgement. So Ana Gallego, she's a PhD student. She has set up all the gut tissue model, and Nerea Sanchez, the tonsil tissue model. Also, Meritxell Genesca, she has been coordinated the tissue network, and also she's doing all the cervix, her team is doing the cervix assays. Vicente Falcón, the chief of the clinical department, also our collaborators in the hospital, pathology, general surgery, otorhinolaryngology, and our collaborators outside. And thank you.